Greetings, everyone. This is Miss Gibson. This is going to be lecture 1 for the immune system. This is chapter 16 in your Med Surg book. It starts on page 316. Here are your objectives. Some definitions that you need to know. Immunity, it's the process in which the body is protected from disease and maintains defense mechanisms. This process requires an interaction of immunity and inflammation to control infection, harmful microorganism, or altered cells. Immunity with inflammation is critical to maintain health and prevent disease. Immunity also plays a role in repair of damaged tissue. Components of immunity would be your intact skin, mucous membranes, and many WBCs and their products. Our bodies can identify self versus non-self. Increased immunity leads to hypersensitivity and autoimmune disorders. Incompetent, this is a term used to describe an adult with fully functioning immune system, maximum protection against infection. All parts and functions are working, maximum protection against infection. They'll have decreased immunity. Decreased immunity can be temporary or permanently endanger one's health. So another definition that you might see is where that they are not, and if they are not immunocompetent, that just means that their immune system is not up to where it should be. So next we're going to talk about some functions of the immune system. Of course, we know it defends and protects, it removes and destroys, and identifies and destroys. So it defends and protects. That's a defense mechanism to prevent re-entry of pathogens. The skin, the skin is a big protector for the invasion of pathogens. Mucous membranes, they secrete a natural chemical to prevent bacterial growth. Our bodies can identify and destroy malignant cells, thereby preventing their future development into tumors. Overview of the immune system. We know that it's activated by minor or major injury. Response may vary depending on the specific antigen or the non-specific antigen, which might cause inflammation. When the inflammatory response is unable to destroy the invading organisms or toxins, the immune system is activated. Self versus non-self. This is just referring to recognition of this is referred to as self-tolerance. Non-self means the body has been infected. Cells either from infected body cells, cells from other people, cancer cells, invading or foreign organisms. We have something called the human leukocyte antigen, or HLA. This is each person's universal product code. It is an antigen on the cell surface. It's a protein on the cell surface capable of stimulating a response. It's present on surface of most body cells, not just leukocytes. It's also called the human histocapability antigen and class 1 antigen. It determines tissue type, which is coded by specific genes inherited from both parents, and it's key to determine self-tolerance. So like I said, the actual definition of HLAs are molecules found on the surface of most cells in the body. They form part of a person's tissue type and play a central role in immune recognition. The HLA system, it is encoded by a cluster of genes on chromosome 6. It is the human version of the major histocapability complex found in other cells, found in other animals. HLA is present on HLA's present fragments of proteins to T cells, allowing the immune system to detect infection, abnormal cells, or foreign tissues. Next, you have toll-like receptors. These are single-pass transmembrane proteins found on immune cells like macrophages, dendrite cells, and also on some non-immune cells. They recognize microbial components like bacteria or viral RNA and initiate an immune response. Toll-like The toll-like receptors are highly conversed across species from fruit flies to humans. So now we're going to go ahead and talk about the organization of the immune system. The immune system is present throughout the body, as we know. Most comes from the bone marrow. Some mature here and others in different body sites. Once the cells are mature, these cells are released into the blood to circulate to most body parts and have specific effects. The bone marrow does produce immature, undifferentiated cells called stem cells. They are pluripotent, which just means more than one potential outcome. And if you look in your book on page 320, figure 16.4, it explains the figure that's on that slide. Next, we're going to talk about leukocytes. These are our white blood cells. Their function is to protect the body from the effects of invasion by organisms. There are several steps there. You have recognition of self versus non-self, destruction of foreign invaders, cellular debris, unhealthy or abnormal cells. Leukocytes can produce antibodies against invaders. They complement immune activation, and they produce cytokines that stimulate formation of leukocytes in, the formation of leukocytes in bone marrow and increase specific leukocyte activity. So this is going to talk about immunocompetence, which we talked about at the beginning. This is 3 processes needed for immunity. If you look on page 320, there's a table 16.2 that goes over each of these. The definition is a protective biological response of the immune system to harmful stimuli such as pathogens, damaged cells, or toxins. It is to eliminate the cause of injury, clear out dead cells, and initiate tissue repair. The vital signs of inflammation would be redness, heat, swelling, pain, and loss of function in the affected area. Next is antibody-mediated immunity. It is the immune defense that relies on antibodies, complement proteins, and antimicrobial peptides found in extracellular fluids. Unlike cell-mediated immunity, which uses T cells to kill infected cells, antibody-mediated immunity targets extracellular pathogens such as bacteria, viruses before they enter cells, and toxins. So the antibody mediated targets extracellular pathogens, where cell mediated immunity uses T cells to kill infected cells. So this is another overview of inflammation. Inflammation is also known as innate, native immunity, or natural immunity. It provides immediate protection against the effects of tissue injury and invaders. It is the first line of defense. The first line of defense is barrier protections, such as your skin, mucous membrane, cilia, urine, gastric fluid, prostate fluid, tears, nasal secretions, saliva. When this is altered, inflammation results. Nonspecific immediate protection against effects of tissue injury and invading proteins and invading foreign proteins are things like it can't transfer from one person to another. There's immediate short-term response, doesn't provide immunity with repeated exposure. Starts with discomfort. It helps to rid the body of harmful organisms. However, if excess response tissue damage is possible, it does help start both antibody-mediated and cell-mediated actions. And then infection is usually accompanied by inflammation, but inflammation can occur without infection. Inflammation does not always mean infection is present. So cell types involved in inflammation. You have neutrophils, macrophages, eosinophils, basophils, and mast tissue cells. The first one we'll talk about is neutrophils or granulocytes. This makes up 55 to 70% of the total white blood count. It's the most plentiful. They are the first responders of the immune system, rushing to the site of infection or injury to fight pathogens. They are produced in the bone marrow and released into the bloodstream. They're also called granulocytes because of the number of granules inside the cell. The different names are based on the shape and maturity. So you have mature neutrophils, less mature neutrophils. The differential of a normal white blood count shows the number in percent of each type. So most are segmented or mature, small percentage are bands. Shift to the left is seen in blood work. That is seen in sepsis. It means more bands than segs present. It indicates bone marrow cannot produce enough mature cells to meet demand, and the bands cannot perform phagocytosis and are of little use in combating infection. Next, we have macrophages. These are released from the bone marrow as monocytes. They mature in tissue as macrophages. Their function is phagocytosis, repair, antigen presenting, processing, secretion of cytokines for immune system control, protect an immediate inflammatory response, and stimulate a longer lasting response. They do have a long lifespan. They can easily detect self versus non self. Their large size is effective in trapping invading cells. Each cell can take part in many phagocyte events. Next is basophils. These are they release histamine, leukotrienes, and other chemicals that can cause allergic reactions. They also contribute to inflammatory responses by increasing blood flow and attracting other immune cells. They play a role in defending against parasites, especially worms. They also interact with other immune cells like eosinophils and mast cells to coordinate a response. Next, we have eosinophils. These come from the myeloid line. They do contain many vasoactive chemicals. They're only 1 to 2% of the white blood cells. They're the most active against infestation of parasites and larvae. They limit the inflammatory reaction. The numbers, they do increase in numbers with allergic reaction. Your tissue mast cells is what we're going to talk about next. They originate in the marrow and do not circulate as mature cells. Mast cells are granulocytes like basophils, but reside permanently in tissue rather than circulating in the blood. They're found in skin, respiratory tract, gastrointestinal tract, and around blood vessels. They appear to be large cells with granules that stain dark purple or blue, similar to basophils. They do release histamine, prostaglandins, and leukotrienes when activated by allergens. They can cause vasodilation, increase vascular permeability, and smooth muscle contraction, leading to symptoms like hives, asthma, and anaphylaxis. There is a defense against pathogens. They help fight parasites, release antimicrobial peptides, and recruit other immune cells. They secrete growth factors that aid wound healing and angiogenesis. They interact with T cells, B cells, eosinophils, and dendrite cells to coordinate immune responses. Next, we have phagocytosis. There are 7 steps to this. This is discussed on page 324, figure 16.8. Goes over each of these steps. So the first step, phagocytes are attracted to the site of infection or injury by chemical signals. This guides them towards the pathogen. Next, the phagocyte binds to the pathogen using receptors like the toll-like receptors. Next, the phagocyte extends arm-like projections around the pathogen. The pathogen is internalized into a vessel called a phagosome. The pathogen is now enclosed in a membrane-bound compartment inside the phagocyte. This isolates it from the rest of the cell. The phagosome fuses with a lysosome, an organelle full of digestive enzymes. This creates a lysosome, where killing and digestion occur. Enzymes like lysozyme protease or protease, and reactive oxygen break down the pathogens. The microbe is destroyed into small fragments. Ingestible debris is expelled from the phagocyte. Antigen fragments may be presented on the cell surface, especially by dendrite cells, to activate adaptive immunity. So like I said, if you want to go into further, if you want to dig a little bit further into these 7 steps, it's in your book on page 324. So next we're going to talk about the stages of inflammation. You have vascular, which is initiation, cellular response, and resolution or tissue repair and replacement. So first you have the triggered event, which is the injury, infection, harmful stimuli, whatever that event may be. The damaged cells release chemical mediators such as histamine, prostaglandins, or cytokines. The blood vessels vasodilate. That increases blood flow. The capillaries become more permeable. Plasma proteins and fluid leak into the tissue, causing swelling. The key player for stage 1 is mast cells and basophils. Number 2 is amplification or the cellular response. This is the chemical signals that recruit circulating immune cells. So the neutrophils migrate to the site and begin phagocytosis. Monocytes arrive and differentiate into macrophages. The complement proteins and clotting factors reinforce defense. Your key players here for stage 2 is neutrophils, which are your first responders, macrophages, and then your complement system. Stage 3 is tissue repair and replacement or resolution. This is pathogen clearance and reduced inflammatory signals. Your macrophages clear debris and dead cells. Anti-inflammatory cytokines suppress further immune activity. Fibroblasts and endothelial cells promote tissue repair and return to homeostasis. Your key players in this is macrophages, fibroblasts, endothelial cells, and regulatory T cells. Next, we're going to talk about specific immunity, which is antibody mediated and cell mediated. So, specific immunity, also called adaptive immunity, is the branch of the immune system that develops a targeted defense against particular pathogens. Unlike innate immunity, which is general and immediate, specific immunity is slower to start but highly precise and provides long-term protection through memory. Your antibody-mediated or humoral immunity, the definition for that is a branch of the adaptive immune system where B lymphocytes produce antibodies that circulate in body fluids to neutralize pathogens and toxins. Works best against extracellular pathogens such as bacteria, parasites, viruses, and toxins. Your cell-mediated immunity is the branch of the adaptive immune system that relies on T lymphocytes rather than antibodies. It is especially important for fighting intracellular pathogens like viruses, some bacteria, and fungi, and also for destroying abnormal cells such as cancer cells. So, With your antibody-mediated immunity, like we said, this uses antigen-antibody interactions to neutralize, eliminate, or destroy foreign proteins. B cells become sensitized to a specific antigen and produce specific antibodies. This begins as stem cells in the bone marrow. They migrate to the second tissue to mature in the spleen, lymph nodes, tonsils, and GI tract. Your antigen antibody interactions have 7 different steps. This is on page 315 if you need to dig a little bit deeper into it. And I've misspoken. It's not page 315. It is page 327, figure 1610. So to go over these steps, with these steps, the bodies learn to make enough of any specific antigen to provide long-lasting immunity against specific organisms. So number 1 is invasion and exposure. A large amount to trigger nonspecific defenses or overwhelm its ability to rid the invader. You have antigen recognition. This is where your macrophages aid by recognizing the invading antigen is non-self and attached to it. The helper cells or inducer T cells together with macrophages bring the antigen into contact with B cells to be recognized as non-self. Step number 3 is sensitization. You have B cells become, a single B cell can only be sensitized to one type of antigen. Immediately after, divides into 2 types of B lymphocytes, both remain sensitized. You have plasma cells immediately start to produce antibodies. Then the memory cell does not produce antibodies until the next exposure to the antigen. Next, you have antibody production and release. This is where antibodies are produced by plasma cells. As many as 300 molecules per second are produced. It's released into the blood and body fluids as free antibodies that are separate from B cells. Next, you have antibody-antigen binding. This is needed for anti-antigen action. Antibodies are Y-shaped and can attach to 2 antigens or 1 in 2 separate sites. The stick of the Y attaches to the white blood cells receptor sites, increasing white blood cells' power as they stick to the antigen. Antibodies may be lethal, but other actions are started, such as neutralization, elimination, or destroying the antigen. Next, you have antigen binding action. This is when it's triggered when antibody binds to when the antibody binds to an antigen. And last is sustained immunity. This provides long-lasting true immunity to a specific antigen when re-exposed. Enough antibodies are produced that illness does not occur. This results from memory B cells made during sensitization. Upon exposure to the same antigen, the memory cells respond by dividing and forming new blast cells and plasma cells. And an antigen is referred to a foreign molecule, often on pathogens, that triggers an immune response. Antibodies or immunoglobulin is a Y-shaped protein produced by B cells that specifically binds to antigens. That definition is important because now we're going to be talking about antibody classifications or immunoglobulins. These are all antibodies. They're globular proteins. They're antibodies that provide immunity. And there are several different types here. If you turn to page 329 in your book, table 16.4 goes over these. You have IgA, which is upper lower respiratory tract to prevent infections. Also can be found in GI and GU tract. You have IgD, which there's low blood concentration in conjunction with IgM. Your IgE is antibody-mediated hypersensitivity. IgG, this is 75% of circulating antibodies. They provide long-term immunity against invading organisms and activate complement systems. IgM is the first antibody formed by newly sensitized B limp plasma cells. 10 to 15% antibodies activate complement system. So the IgM is the first antibody formed by sensitized B cells. That's all you need to know about IgM. Once again, that's in your book on page 329. So acquiring antibody-mediated immunity. The body learns to respond to invasive organisms or foreign proteins. You have active immunity. This is where the antigen enters the body, responds by making specific antibodies against the antigen. Active is body takes an active role in making the antibodies. You have natural active immunity. This is where the antigen enters the body naturally without human assistance, often causes diseases first exposure. It's most effective and long-lasting. Then you have artificial active. This is your vaccinations, immunizations, attenuated antigens such as tetanus, polio, diphtheria. Sometimes you may need a booster. But they're given to less likely cause disease formation of antibodies. Then you have passive immunity. This is antibodies are transferred from another human or animal. This is short-term protection. You have natural passive, which is passed from mother to fetus or infant through placenta or colostrum and breast milk. You have artificial passive, which is immunity to prevent disease or death when exposed. And an example of the artificial passive, I cannot talk, is when you're bitten by an animal that might be rabid, you have to get the rabid vaccine. So, or the rabies vaccine, or tetanus is another example of that as well. So next we're going to talk about cell-mediated. This involves many white blood cells, actions and interactions. Cell types is you have helper and inducer T cells. They act as organizers. They recognize non-self and then secrete cytokines that enhance activity of other white blood cells and increase overall immune function. The organizer or calling in the arm squad of WBCs involved in inflammatory and antibody and cellular production. The suppressor T cells, they act as regulators, have opposite function of helper cells. They prevent hypersensitivity when exposed to non-self or protein. Next, you have the cytotoxin T cells. They destroy cells that contain human leukocyte antigens, most effective against parasites, allow an immune system to recognize the infected cell as abnormal. Next is the natural killer cells. This is very important in cell-mediated immunity. They seek and destroy non-self cells, most effective in destroying cancer cells or viral infected cells. Your cytokines, these are small protein hormones produced by many white blood cells and some other tissues. There's two types, monokines, which are made by the macrophages, neutrophils, eosinophils, and monocytes, and then lymphokines, which are made by the T cells. They act like messengers that tell specific cells how and when to respond. Cells that change their activity when a cytokine is present are responder cells. Your cytokines include interleukins, interferons, colony stimulating factors, tumor necrosis factors, and transforming growth factors. Protection provided by cell-mediated immunity helps protect the body through the ability to differentiate self from non-self. Watches for and rids body of self cells that may potentially harm the body, and it is important in preventing the development of cancer and metastasis after exposure to carcinogens. Age-related changes in immunity. As we get older, the increased risk of autoimmune and infection increases. The normal flora of our skin becomes the function of the normal flora of our skin is decreased. There's a decreased number and function of neutrophils and macrophages. The older population may not have a fever, even with severe infection. Less recognition of pathogens increases the risk of sepsis. Increase in white blood cell counts may be delayed or even absent. T cells may remain constant but less functioning. Takes longer to develop antibodies. Your memory cells are slower, may need boosters. Loss of recognition of self, increasing autoimmune responses. So those different stages that we've already talked about with inflammation, you're going to have decreased neutrophil function. Leukocytosis doesn't occur during acute infection. Older adults may not have a fever with inflammatory or infectious episodes. For your antibody-mediated immunity, there's going to be a decreased number of forming B lymph, and ability to develop into antibody-secreting cells are decreased. Older adults can't develop new antibodies, thus needing vaccines such as the flu, pneumonia, shingles, and COVID vaccine. Cell-mediated immunity, number of circulating T cells decrease. The older generation is more at risk for bacterial and fungal infection, especially on the skin, mucous membrane, respiratory tract, and GI tract. So now we're just going to go through some practice questions. Risk for infection. So we said it shows a shift to the left. Make sure you go back and look over what I said was effective when we're looking at the white blood cell count and there's a shift to the left. What's happening here to cause that? the bone marrow. We know that immune function is most efficient when people are between the ages of 20 and 30. With infancy and your teen years, the immune system is developing and changing. As we get older, everything declines. So 50 years and older, your patients that are 50 years and older, the immune system is beginning to decline there. So what did we say? Think back to what we said caused the pus. Responses at the tissue level can cause the 5 cardinal symptoms of inflammation, which are warmth, redness, swelling, pain, and decreased function. Stage 2 is the cellular exudate part of the response. In this stage, neutrophil, an increased number of circulating neutrophils occurs. Exudate in the form of pus occurs containing dead white blood cells, necrotic tissue, and fluids that escape from the damaged cells. Here are some key terms that you'll need to know for your test, so make sure that you know those and understand what they are. If you have any questions, please don't hesitate to email me. Have a great day.